Okay, so good evening. Let's start today's session of Enzyme Science and Technology. So, in today's session, we'll be discussing few questions that are related to the application of the enzymes. So, till now, we have studied many enzymes, how they function, what are their properties, their inhibitors, their kinetics, and everything. Now, let's study why these enzymes are important, why even their production is important, what are the use of those enzymes. Okay, so for that, let's start. So, enzyme, we know, have several applications in our daily day-to-day life. So enzymes are involved in medicinal industry, in food industry, and in agriculture, in genetic engineering, and environmental related uh, properties. Okay, so in medicinal also, enzymes can be helpful as a diagnostic tools, as a diagnostic markers or tools, and also for drug development or drug delivery. Okay, in food industry also we have several different uses of enzyme. For example, in poultry industry, in fish production, and in wine and juices, these kind of liquid and juice production. In genetic engineering also. Okay, and in agriculture also there are several uses of the enzyme. So we will be talking mostly about those enzymes and their functions. So uh, if we'll talk about the food industry, it can be divided into several different parts. For example, we use enzymes in dairy. Can you please mute yourself? Okay, so here we are talking about the applications of enzyme, and I have mentioned that enzymes are involved in medical field, in food industry, in agriculture, in genetic engineering field. In food industry, we have poultry, fish, wine production. In medicinal, also we have the use of enzyme as a diagnostic tool or for drug delivery or drug development. So there are several uses of the enzyme and several applications. So in food industry, if we'll talk about, we have dairy industry, and we have brewing industry, baking, and wine or juice production, wine or juice production, and also meat production. Okay, so if we talk about dairy, so there are several enzymes that are being used in dairy field, such as rennet, which converts uh, milk to cheese. Okay, we have lactase enzyme. Okay, which uh, which is used in the processing of milk. So this lactase break down the lactose sugar, and that is why it helps us to produce lactose free milk products. Okay, and we also use proteases and catalases as in to prevent the oxidation. Okay, these are the dairy industry enzyme. Also we have in brewing we have alpha amylase, we have proteases, we have amyloglycosidase. baking industry, uh, in baking industry, the enzyme that are being used are the glucose oxidase or maltogenic amylase. It is one of the very important enzyme which is used in the baking industry, which is maltogenic amylase, which is used as anti-stalling reagent, so that the breads and buns which are prepared in the baking industry, they remain moist and doesn't get uh, hard or very, it should be in a moist condition, in nice and soft condition. That is where in the baking industry this enzyme is used. And then we have wine and fruit juice producing industry, where pectinase and beta glucanase enzyme is also used. Okay. Pectinase. In meat industry we use proteases and papain enzyme for the tenderization of the meat. So papain and protease break the bonds that are present in peptides. As we know, in the meat we have the muscles where we get the protein from. So to make the meat more tender, these enzymes papain and proteases are being used so that it breaks the bonds between the protein, so that the meat can become more tender. Okay, so let's start today's question. If you have any doubt, you can ask me. So, yeah. So the first question here is, which of the enzyme is used in the production of cheese? Just now we have talked in dairy industry, we have rennet enzyme, which is used to produce cheese from milk. Okay. So the correct option will be rennet. Yeah. So which one of the following is used to tenderize the meat? Okay. So who will like to answer? Anybody? Anyone? Which enzyme is used to tenderize the meat? I just now told. What would be the correct option? Okay, no one is trying to answer. Okay, so for tenderization of the meat, the enzyme that is being used is papain. Okay, next we have, which of the enzyme is used in the removal of starch from beer and fruit juices? Okay, so amylase is an enzyme which is converts starch to dextrins and producing corn syrup. Okay, so for the processing of starch, the any content which contains starch in their particular forms, there this particular enzyme amylase is being used. Okay, and that catalyzes the starch content of that particular mixture. So, okay, so these uh, starch is obtained from most of the microorganisms, plants, animals that possess the ability to utilize the starch as a carbon source. Okay, for example, in bacteria, fungi, yeast, everywhere this amylase enzyme is present. Okay, and there are for the many industries that are producing this amylase, because it has very much use in the beer and fruit juice industry. Okay, so wheat barn is used for economic production of alpha amylase. Okay, wheat bran. Okay, so this is used for the production of alpha amylase either by solid state or liquid surface fermentation process. Okay, and there are reagents which is cholic acid and twin 20 or twin 80, twin 20. So these are some harsh detergents. Okay, twin 20, twin 80 and cholic acid. So these are used to increase the production of alpha amylase by 2 to 3 fold by increasing the permeability of the cell membrane. Okay, and Let's go to the next question. So yeah, so which one of the enzyme has anti-stalling effect? Okay, I just now told in the baking industry. Okay, so anti-stalling means to maintain the moisture and the softness of the product. Okay, for example, in breads and buns in the baking industry, these enzymes are being added so that after the baking, when it goes to the natural temperature normal environment, it doesn't get solidified or hard. So to maintain that moisture and that softness, this anti-stalling enzyme has been used, and that anti-stalling enzyme is maltogenic amylase. Okay, so that is the correct option here. Yeah. So this is the question, like which of these increase the production of alpha amylase by increasing the permeability of cell membrane. Okay. So alpha amylase that we talked, that is used for catalyzing the starch. Okay. So this alpha amylase, for the production of this alpha amylase enzyme, which are the reagents that being used for increasing the permeability of cell membrane. So we have four options: 20, folic acid, both, or none. So anyone like to answer? I just now told. Okay, so I just now mentioned that 20 and folic acid both are used to increase the production of alpha amylase in the industries. So the third option will be the correct option. Next, so this alpha amylase is a metalloenzyme. So metalloenzyme means that this enzyme needs certain ions, divalent metal ions, to perform its activity. So this alpha amylase is also one of the metalloenzyme, and it contains at least one calcium ion. Okay, so the correct option will be calcium ions. Okay. So this question is, soya sauce can be produced by the action of which enzyme? So here for the soya sauce production, proteases are being used extensively. Correct option will be proteases. This is an enzyme used to dissolve blood clots. Okay. So which enzyme is used to dissolve the blood clots? Anyone like to answer? Maybe I need to check it once again. Be able to tell. So here is the question is, which of the following enzyme is used in the treatment of cancer? Okay. So for the blood clot dissolving, the correct option would be streptokinase enzyme, which is added in the next option. Error. So streptokinase is used for the blood clot dissolving. Okay. And here, so here we have, which one of the following enzyme is used in the treatment of cancer? So one of these enzyme, out of these four, one enzyme which is be used as a treatment of cancer. So this enzyme is used as a part of chemotherapy. Chemotherapy. Okay. So in case of leukemia, which is the blood cancer, Okay, so in leukemic cells, so those cancer cells have don't, unlike all the other cells of our body, which has their own machinery to synthesize the amino acid asparagine. Okay, so but these leukemic cells, they don't have the machinery to synthesize the asparagine, so they utilize several different pathways and produce the asparagine. So this uh, but in chemotherapy, asparaginase is the enzyme which is used so that the asparagine produced in the cancer cells can be degraded. So in other cells, as they have the machinery to produce, they can produce more, but the cancer cell doesn't have the machinery. That is why these asparaginase degrade the produced asparagine and helps to cure the cancer. Okay, so this is the correct option, which is asparaginase is an enzyme which is used as a treatment for cancer and used in a chemotherapy. So asparaginase will be the correct option here. Next, we have pectinase can be derived from the source S. Okay, so what does pectin do? Okay, so pectinase enzymes, these are used for extracting juice for puree formation. Okay, so this enzyme pectinase break down the substrate pectin and then the juice is extracted. So uh, So it is an integral part of the commercial food processing industry where it is used to degrade the pectin and facilitate different processing steps such as liquefaction, clarification, and juice extraction. Okay, liquefaction, juice extraction, juice extractions, and clarifications, juice processing. Okay, and the substrate of this enzyme is pectin. Okay, so the source of this pectinase is from the citrus fruits. We can get the pectinase from the citrus fruits. So these are the main source for producing the pectinase enzyme. Yeah. So which of the following is not an application of pectinase? So pectinase is used um, juice clarification, uh, wine and juice industry. There also it has been used for the clarification purpose, and in also in the coffee fermentation process and chocolate production process also this pectinase is used to break down the pectins. So here also it has been used, but it is not being used in the recycling of the plastic, so this is the wrong option here. So this will be the correct. So yeah, so which one of the following is not an inhibitor of alpha amylase activity? Okay, so alpha amylase we just now discussed is used as a starch uh, uh, catalyzing industry. Okay, starch processing industry for the hydrolysis in the production of starch hydrostates. Okay, and it is also being used in the removal of starch in beer and fruit juices, or from clothes or from porcelains. Okay, so this enzyme is having like heavy metals, cations, sulfhydryl group reagents like these. So these are the common inhibitors of the enzyme. However, this chloride ion is not an inhibitor of the enzyme. It is actually helping in the activity of the enzyme. But the other BSA, N-bromosuccinimide and EDTA are the inhibitor of the alpha amylase activity. Okay. So correct option will be this fluoride, which is not an inhibitor of the alpha amylase activity. So which one of the following agents has been reported to have inhibitory effect on the production of enzyme? So here the correct option will be calcium ion. So calcium ion has a inhibitory effect on the production of alpha amylase. However, uh, twin 80 and folic acid, as I have mentioned, it is used to increase the production, and similarly like glycine also. And but the calcium ion, if it is added, it inhibits the production of the alpha amylase. So correct option will be option C. Okay, so these were the all questions which I have selected today. These are very direct answering questions. You just need to go through the notes and all those informations will be there. So the question which I have taken here, these are the kind of question which can appear in the exams of this course. So if you have anything to ask, you can ask me. And these were all the questions for today. If you want to ask me anything, you can ask me. Otherwise, I'll conclude the session. Okay, if there is nothing to ask, should I conclude? Okay, so I'm concluded today's session. See you in the next class.